Last topic, receptor-mediated endocytosis. This is the subtype of active transport. The receptor-mediated endocytosis, lot of receptors are present for endocytosis. By this mechanism, protein can absorb, polysaccharide can absorb, iron can absorb, LDL, low density lipid can absorb. In this case, the ligand is bind with the protein, and that complex is then bind with the receptor, and then this complex and the receptor are internalized. When they are internalized, they are digested. The protein, the receptor are recycled. They are recycled cell membrane, and the ligand is digested. So this is known as receptor-mediated endocytosis. For example, this is iron. Iron bind with ferritin protein in the blood. This is iron ferritin complex. This iron ferritin complex is then binding with the ferritin receptor. Once this complex is bind with this ferritin receptor, the calithrin protein pulling this area toward the cytosol. This pulling is in progress. This is in progress until this area is internalized in the form of a vesicle. Now this vesicle is calithrin coated vesicle. Again calithrin coated vesicle. Lot of vesicle are internalized by this way. ये आयरन और फेरेटिन जब आपस में बाइंड हो जाते हैं तो फिर ये कॉम्प्लेक्स इस रिसेप्टर के साथ बाइंड होता है. फिर जब ये रिसेप्टर के साथ बाइंड हो जाए तो अंदर की तरफ सेल मेम्ब्रेन के इनसाइड कैलेथ्रीन प्रोटीन होते हैं. ये कैलेथ्रीन प्रोटीन इसको खींचते हैं अंदर की तरफ, इस तरह खींचते हैं. अब इसको खींचते अंटिल कि इसको एक विजिकल में कन्वर्ट कर ले, और इस विजिकल के इर्द गिर्द कैलेथ्रीन प्रोटीन होते हैं. अब बहुत सारे विजिकल अंदर चले जाते हैं. सो द विजिकल आर फ्यूज्ड विद ईच अदर. दिस दे आर फ्यूज्ड. नाउ दिस इज फ्यूज्ड नोन एज प्राइमरी एंडोसाइटोसिस. दिस इज नोन एज प्राइमरी एंडोसोम and this is secondary endosome. When they are fused, the internal environment of this primary endosome is acidic. This acidity digesting the receptor and iron ferritin complex. After digestion, the receptor are separated from the iron ferritin complex. Just like look here, receptor are in one vesicle and iron ferritin complex are in next vesicle. They are detached by the lysosome enzyme, and these receptors are again recycled and transplanted at the cell surface and working again. Now this ferritin iron complex is digested. Iron is get free into the cytosol for biological action.